Thank you, everyone. Thank you, everybody, for being with us today. And it's my pleasure to introduce Professor Elva Diaz. She did her undergraduate studies in biochemical sciences in Harvard College, and graduated from her PhD in biochemistry in Stanford University, and she then did a postdoctoral research trainingship at UC Berkeley in developmental neurobiology. And since then, she has been a professor at UC Davis, and her lab focuses in the molecular mechanisms of excitatory synapse development, function, and plasticity in the central nervous system. And today she's going to talk to us about how to change your mind, the molecular mechanisms of brain function, underlying learning, and memory. Thank you so much, Elva, for being here with us today. Go ahead, please.
Thanks, Veronica. Always a pleasure. So hi, everyone. Those I know, those I don't know, or those I haven't met in person. There's still several people we haven't been able to meet in person yet. As you know, these Campos presentations combine research as well as individual background. I like to use these two questions to build community amongst my colleagues. The questions are, who are you and why are you here? And so I'll attempt to address these questions as I start the presentation. Who am I and why am I here? Oops, how do I go forward? So this is an example of a cross-section through a rat brain from the Allen Brain Atlas. And the different colors represent different transcripts of genes that are expressed in the nervous system, at least in the rodent, as a model for human. And when I was a graduate student, I went to a Gordon conference called Cell Biology of the Neuron. I wasn't a neuroscientist at the time, but I went to this conference because we were studying receptor trafficking in non-neuronal cells. And there were a lot of similarities with receptor trafficking and vesicular transport, for instance, during synaptic vesicle transmission. And it was there that I was first exposed to the complexity and the amazing molecular and cell biology of the nervous system. I'd actually never taken a neuroscience course ever. It's kind of ironic that I'm the chair of the neuroscience graduate group, having never taken a course before. Again, my background's in biochemistry. But I was fascinated by the structure. And when I started grad school in the early 90s, it was such a revolutionary time in biology and in the environment in general, right? The internet was just taking off. When I started graduate school, I remember distinctly that the chair of the program said, "Oh, we're going to give you all email addresses. Be sure you check your email once a week because we send out important notices to email," right? Now, if you don't check your email every couple of minutes, right, how you're way behind. And then also the internet, right? So this also was developing in terms of becoming mainstream. And this really facilitated research, biomedical research. And I remember as a student doing a yeast-2 hybrid screen for the first time, identifying my candidate proteins candidates, and then isolating the cDNA that encoded the proteins, and then sequencing them myself by hand, right, with radioactivity, reading off the sequences, and then going to the one lab in the entire department that was connected to the internet because they did computer modeling of protein structures. So one computer attached to the internet in our whole department. And I remember typing in the sequence of these candidates that I had identified, and talking to the graduate student who said like, oh, there's this new tool called BLAST. And we typed in my results of the candidates that I had and pressed the enter button. And he said to me, come back tomorrow morning and we'll have the results, because that's how long it took to do a search on the internet, hours instead of, seconds. So whenever I look back about, where I am now, it's always, I find interesting to see where I started. And again, what I became fascinated by, all of us are driven by the questions in science, was the concept that the brain is the most complex organ in our body. It contains approximately 100 billion neurons and glial cells. And each of these neurons can have 1,000 or 10,000 synapses. So this can then lead to trillions of synaptic connections. And this is how our brain cells communicate. And there's estimates of hundreds to thousands different cell types. So at the time, with the emerging genomic era, the internet, the tools that were available, I was really inspired by trying to understand, taking a molecular approach to understanding brain development and function. And in particular, the fact that out of the 25,000 or so genes that are found in the human genome, many are either exclusively expressed in the CNS, potentially a third are exclusive to the CNS, but at least close to 80% are expressed somewhere in the nervous system. So to me, this is just an amazing, enormous complexity within this one structure of our organisms that, again, gives rise to our amazing ability to remember, to learn, to think, to be conscious. All of these things, I have found the passion that instills in me. And as a biomedical researcher, I find, or at least when I write grants, as we all do, right? CNS specific gene products could represent then novel therapeutic targets for neurological or psychiatric diseases. So as I mentioned, this is a cross-section demonstrating some of this complexity. So these transcripts are in different colors, and you can see, for instance, this, let me get my pointer. The pink gene is expressed here in striatum and parts of the cortex. You have the cyan expressed in the dentate gyrus of the hippocampus. These are different structures in the nervous system. And so it's not a homogeneous tissue. We've been focused on the hippocampus. Like many researchers in the field, the hippocampus is a structure critically important for cognitive abilities such as learning and memory. And in particular, we've been focusing on the individual neuronal cell types within the hippocampus, which are pyramidal cells. And as I mentioned, when I went to this Gordon conference on cell biology of the neuron, also in the early 90s, this was at the time when the ability to culture primary neurons from rodents was developed by Gary Banker and colleagues at Oregon Health Sciences, OHSU, or University of Oregon. But this method that allowed researchers to isolate embryonic tissue from rats or mice and then dissociate them into single cells and plate them in dishes and allow them to develop. And this is an example of a single cell. The pyramidal shape is where these neuron types get their name. And they're labeled in a couple of different markers. So the red is a marker of dendrites, which is labeling MAP2, a microtubule associated protein. And the green is a synaptic protein that we identified and have been characterizing in the lab. And as you can see, these dendritic structures protrude off the dendrite shaft. And it's these sites that form synaptic connections with axons from other neighboring cells. So this technique, again, pioneered by Banker. In fact, these dissociated hippocampal cultures are often referred to as the Banker culture, representing the development by Gary Banker and colleagues. And he was very generous in allowing people to come to his lab and learn how to do this technique. So it quickly disseminated into the field. As I mentioned, I was in biochemistry studying receptor trafficking in non-neuronal cells, but I remember this system from when I was an early stage graduate student. And then when I decided to switch fields as a postdoc, I went into the molecular and developmental neuroscience to try to combine cell biology of the neuron and trying to understand this functional complexity of the different types of genes and gene products expressed in the brain. So I like to think of this as it's almost like a circuitous journey. You know, as I mentioned, I never had taken the neuroscience class. Maybe had I taken neuroscience as an undergraduate, I would have been fascinated earlier with this field and moved into neuroscience sooner. But I really appreciate my foundation and fundamental training within biochemistry. So I'm a native Californian. My parents are originally from Mexico. They immigrated to the US, California, Central Valley when they were kids with their parents. And they were first generation college students. They were able to get into the education system. They both attended San Jose State and became high school teachers. My dad eventually became a real estate agent. I think he didn't like the teaching aspect, too much paperwork, he would always say. But my mom was a lifelong teacher, and that made a huge impact on me in terms of lifelong learning. So the expectation was that me and my 3 siblings, I'm the oldest of 4, that it wasn't if we were going to go to college, it was when. And so that was never a question about whether I would go. It was just when, you know. And like a typical child who rebels against their parents, I remember having arguments with my mom saying like, I'm a straight A student. I should be able to stay out all night. And she would never agree with me. So we had our differences in opinion, but my mom was special. And some of you, at least I think, Veronica, hopefully you met my mom. Some of you have met my mom. She passed away in 2015. But she was very special. And part of the reason I went so far, if you notice, the only time I left California in my entire life was to go to college on the East Coast. And part of it was rebellion. And you could think of it as like, okay, yeah, going to a place like Harvard, that's rebellious. But it was my sort of like need to get away and be independent. And I must admit, I think that was good for me because going to college at a place like Harvard was really challenging for me. I didn't realize how limited in resources my high school was. It was reasonable, but it wasn't at all, it was below average. You know, I had below average scores on the SAT. There was below average number of, you know, we had like 2 AP classes and things like that. Everyone had all this like prep work. It was things I had never heard of. And that's also part of the reason that drives me as a mentor, especially for students here in California who have similar backgrounds as myself, but maybe didn't have the resources or the privilege that I had in order to do well during high school and college. Because of my parents, hard work and work ethic they were able to provide for my college education. I had a part-time job, but it wasn't that I was needed to work to support myself. My parents were able, because of their hard work and their parents' hard work, to provide for their children. But that's really motivated me to give back to my community, especially here in California, where so many people have a similar background as myself, but have not had the privilege or the resources. So it also, this time away from college, led me to really appreciate the diversity, the progressiveness here in California. And since that time, I came back immediately to the Bay Area. I was within about 30 minutes of my mom and dad, which was great. I miss them so much. And then, of course, staying in the Bay Area for a postdoc. And I feel, again, so fortunate to have been able to get a position close by at UC Davis. And so I still have two siblings, two brothers who live in San Jose, and then my sister, who lived in New York for, I don't know, 20 years, just recently moved to Australia. Her husband's Australian, but she comes back to visit. And of course, I'm looking forward to visiting her. So all of these things, like I said, I feel like I had the ability to explore during my training, to discover my passion in terms of understanding the cell biology of the neuron, because of the privileges and resources and the support that I had growing up from my parents and from colleagues and friends. And that, again, motivates me to give back to and to mentor students at present. And so I again, feel very fortunate to be at a place like UC Davis, where I've even encountered multiple students who've gone to my high school. And there was even a couple of students who had my mom as a high school teacher, years ago, because she retired in the early 2000s. So that was really special to be able to meet an undergraduate student here who said, like, oh yeah, I had your mom as my, Spanish teacher, Señora Diaz. Everybody loves Señora Diaz. So that is a lot of who I am. My family, my support network really motivates me, not only for my own passion in research, but passion in mentoring. Okay, so I'm going to tell you a little bit about some of the work that we do in the lab, and feel free to ask any questions. Again, this is meant to be more of a conversation. So as I mentioned, we use this system, this dissociated hippocampal culture system, to allow molecular and cellular inquiries of how neurons work, right? How do they specify their connections? How do they communicate with other neurons? And then ultimately, how do they impact neural circuit behavior so that we can do things like learn and remember? And so if you zoom in to one of these synaptic structures and then turn it into a cartoon, you'd see something like this. So this green part here is the dendritic shaft, which would be represented in the red structure here. And then the protrusion, this green protrusion, this is what's called a dendritic spine, which protrudes off the dendrite. It's thought to represent a compartment within the neuron, so that you have this single unit of connection between a presynaptic axon from a different cell that can then synapse through these transsynaptic adhesion proteins in order to generate efficient synaptic transmission. So this is our synaptic vesicle, which would be filled with neurotransmitter, and upon an action potential, signal would release and then bind to neurotransmitter receptors on the postsynaptic side, such as AMPA receptors and NMDA receptors. And so we've been studying at the molecular and cellular level how this synapse structure forms, how it knows to form the right structure with the appropriate target, and then how synapses can be strengthened or weakened, because we know that synapses can constantly undergo different types of plasticity as we're learning. The other reason I find synapses very fascinating is that a wide variety of neurological and psychiatric diseases are associated with deficits in synapses. And so here's an example, again, of one of these pyramidal cells. You see the processes coming off here. And here's an example with the schematic of these dendritic spines, the sites of postsynaptic input. And in mechanism or in disease states such as autism or Rett syndrome, you can see alterations in the structures, in the number of spines. So autism is thought to involve an exuberant number of synaptic connections, and individuals are challenged then by this overactive network. In contrast, such as Alzheimer's disease, one of the first things that happens during the loss of the during the disease state is loss of synapses, and then ultimately the entire neuron will degenerate. And you can see different types of phenotypes with these other disorders. The other reason it's very fascinating, I just finished my last teaching responsibility for the medical school. So I'm one of the course directors for the Brain and Behavior course for second-year medical students, in addition to teaching molecular and developmental neuroscience with Crystal, who's on the Zoom call. For graduate students, I teach neuropharmacology to medical students at UC Davis Health. Question?
Hi, sorry for the delayed question. I was just interested, do you know which genes are exactly involved in the differences in synapses compared to like the normal brain and the other psychiatric diseases?
Yeah, so that's a great question. There's two major classes for, and you're asking about the disease genes, right? So genes associated with disease, yeah. So there's two major classes. There's genes that are involved, one class of disease state, of genes that are involved in disease states are these transsynaptic adhesion proteins, so mutations in certain family members that affect these adhesion contacts. There's also mutations known in proteins within the scaffold, this postsynaptic density structure, which is a scaffolding compartment that holds everything together. Some of the most prominent disease mutations in these disorders affect this scaffold and these connections. And then secondary to that, a second class of molecules is involved in chromatin remodeling. So presumably something in the nucleus, and that's not listed here. So two separate classes, they could be related, right? So maybe the chromosome remodeling disease genes are impacting the expression of some of the synaptic proteins, or perhaps the synaptic proteins, in addition to building the structure, signal back to the nucleus for certain forms of plasticity. And so those are the two major classes that are thought to underlie these disease synaptic phenotypes associated with disease.
All right, thank you.
And so in the neuropharmacology course that I teach for second-year medical students, I also highlight that the vast majority of the drugs that are used in the treatment of neurological and neuropsychiatric disorders, such as schizophrenia, epilepsy, Parkinson's disease, bipolar, antidepressant, all affect to some degree aspects of synapse function. So for instance, you have molecules such as, there's my standard over here. No, I don't have it. Where's the blocker? Well, so for instance, like there's molecules that block the release of neurotransmitters. There's drugs that block the postsynaptic neurotransmitter receptors. There's drugs that can affect metabolism. So understanding basic mechanisms of synaptic transmission can also help to understand or identify novel targets of for these disorders. And then finally, as I mentioned, plasticity. So here, this is a very simplified version of a single synapse. For instance, again, with this postsynaptic side, the presynaptic side that's joined by these transsynaptic bridges. But what's amazing is that this structure has to change during the course of development, not just when it's being built, but also as we're learning. So for instance, the AMPA-type receptors, do I have it better? No. So the AMPA-type receptors, which are schematized here in green, correlate with how strong a synapse is. So if you have more AMPA receptors in your synapse, you will have a stronger synaptic connection. And so these AMPA-type receptors are constantly altered depending on the state of the circuit. So you can easily, not easily, but you can remove or add more of these AMPA receptors during certain states of learning and plasticity. And and this is where I was talking about receptor trafficking in neuronal cells. I was studying receptor trafficking in non-neuronal cells. For those who study any form of cell surface receptors, you know, receptors have to be synthesized in the ER, go through the secretory pathway to get to the plasma membrane, but then they can also be endocytosed. And in the case of synapses, this is what underlies whether a connection will be strong or weak. And so there's classic types of synaptic plasticity, which are called LTD for long-term depression, or LTP for long-term potentiation. And the idea is that under certain forms of activity that the synapse interprets as being strengthening or to lead to strengthening, they will automatically, very quickly incorporate more AMPA receptors into their synapse. And then in cases where the signal is interpreted as a signal to weaken synapses, they will remove AMPA receptors. So trying to understand the mechanisms for how this particular receptor type is trafficked has been a decades-long investigation in the field of synaptic plasticity. As a cell biologist, molecular biologist, biochemist, again, I'm very fascinated by how these receptors can move around. And from a basic cell biology point of view, like I mentioned, you have surface receptors that can laterally diffuse in the fluid mosaic of the biological membrane. They can be removed. They can undergo endocytosis. They can be transported to an intracellular compartment called the endosome, where they can either be recycled back to the plasma membrane or targeted for degradation. And all of this happens continuously in the lifetime of neurons, because unlike other cells that can replenish themselves throughout our lifetime, such as our skin, our hair, our blood, the neurons that we were born with are basically the only ones we're going to get. There's two areas of the brain that have been demonstrated to continually generate new neurons. Whether and how much that occurs in humans is still an ongoing question. But the vast majority, 99% of the neurons that we have in our brain now, were the ones we were born with, or the ones we developed soon after we were born. And again, from a cell biologist point of view, this to me is just fascinating that it's these same cells that have been able to change over the course of decades of our life, right? Okay, so the AMPA receptors is what we've been focusing on, trying to understand how this particular type of receptor can traffic to synapses, how it might be removed from synapses, the sorting mechanisms that regulate this. And then also, it turns out there's a wide variety of interacting proteins that associate with the AMPA receptors and can dictate not only their function, but their localization, whether they get targeted for endosomes or get targeted for the postsynaptic density. And so we've been trying to understand some of these molecules that associate with AMPA receptors to try to understand how synapses can be strengthened or weakened. This is an example of in this case, this was a crystal structure. This was before the cryo-EM revolution. This is a crystal structure of the tetramer of the AMPA receptors, which are composed of 4 subunits, GluA1, 2, 3, and 4. And each of the different subunits is color-coded here to demonstrate the different polypeptides. And you can see here's the transmembrane domain, and then it has these very large extracellular regions. And this is where you get binding to the neurotransmitter to open or close the channel. Because, and I guess I didn't mention this, so these, as I mentioned, AMPA receptors are important for excitatory neurotransmission. So it's been difficult to develop therapeutics for against the AMPA receptors for things like epilepsy, which is due to excessive excitatory neurotransmission, because this is important for our baseline activity, right? We wouldn't want to inhibit our baseline activity. However, there have been a couple of non-competitor AMPA receptor antagonists that were developed. But a recent area, as I mentioned, has been to try to focus on these auxiliary subunits that associate with AMPA receptor. So biology is amazingly complex. As I mentioned at the beginning, there's this huge amount of gene expression, diversity of gene expression that exists in the brain. And in fact, these AMPA receptor subunits, so I've taken this, what we're looking at is this structure and turned it 90 degrees, and now we're looking at it down. This AMPA receptor associates with a wide variety of unrelated proteins. So these are all listed in these different names. Again, the names aren't important except that they are reflecting this diversity of gene expression. And many of these are expressed in only certain cell types in the brain or certain areas. And so now we have a mechanism by which we can get some specificity to the AMPA receptor complex, both in terms of its ability to regulate synaptic strength, but also for therapeutics. And so, for instance, there have been a recent, here's an example where targets, novel therapeutic targets, again, for chronic pain or epilepsy in this case, aren't targeting the AMPA receptor itself, but instead are targeting one of these associated proteins as a mechanism for specificity. So this is really exciting from a therapeutic point of view, and also for us, this might be a way that we can start thinking about possible novel therapeutics. Okay, so let's see. So we've been studying one of these auxiliary factors in terms of trying to understand mechanisms of AMPA receptor activation, plasticity, trafficking, and function. And so again, using, as I mentioned at the beginning, I was interested in gene expression profiles in the brain. and did some early gene profiling, expression profiling studies of the developing brain, and identified candidates that we could then study the function of in the laboratory. And one of these molecules we identified, we named SynDIG1 for Synapse Differentiation Induced Gene 1. So this gene was upregulated during the development of synapses, and it turned out to be an important AMPA receptor interacting regulatory protein. It is a founding member of a superfamily, like of a family of genes, like many are in the nervous system, and actually throughout or the complex organisms. And I'm going to tell you a little bit more about what we've been doing recently with a related protein, SynDIG4, which is also referred to as proline-rich transmembrane domain, because it has a large number of prolines in its primary structure. Okay, and then as I mentioned, this is the AMPA receptor structure, and this is with one of its other AMPA receptor auxiliary factors. The name is called stargazin because it was identified originally from a mouse mutant that was called stargazer. It had a mutation in this molecule that, when mutated, the mice would have these absence-like seizures, look up to the sky, hence the name stargazing. And so there was a recent paper last year from Eric Guo at the OHSU that identified SynDIG4, PRT1, that we study as being intimately associated with the AMPA receptor complex that contains two other auxiliary factors. And this was endogenous hippocampus AMPA receptors. So this is demonstrating that this molecule that we've identified, that we've been characterizing, is in fact associated with endogenous receptors. And this is, that's like to demonstrate a little bit more, you can somewhat see this is the transmembrane domain. The two of the subunits are shown here for the AMPA receptor, and there's this very tight association in the transmembrane domain of SynDIG4, Hornischan, another auxiliary factor, and the stargazin or TARP. Okay, I'm just going to give you a few of the highlights from a publication that was that we published a few years, actually now it's been almost 5 years ago. But this also highlights, I think, some of the strengths of UC Davis that I'm sure many of you are aware. The fact that Davis is a highly collaborative and collegial and supportive environment. So we've collaborated with my department colleague, Johannes Hell, for many years. And then also Jackie Crawley, who's at the Mind Institute, or she's retired now, but again, to do behavioral analysis. And this has been great because anytime you need or want to do a new approach or technique, it's always straightforward and easy to find a colleague at Davis. And that's been one of the most satisfying things being on faculty here. So I'll give you a little bit of the highlights. So in molecular biology, one of the things when you have a molecule and you have an idea of how, or you want to test how it's impacting your question of interest, in our case, synapse function, you can generate a transgenic mouse in which the gene is knocked out. And doing this, we found that the synapses of the SynDIG4 knockout animal were inhibited in their ability to undergo LTP. And this led to very impressive cognitive deficits that I'll show you just in a moment. This is a schematic of how you make this genetic knockout. In this case, we've removed the locus for SynDIG4 and replaced it with a LAC-Z, and this allows us to be able to see expression of the gene promoter, which is highlighted in blue here. And then we looked at function, like what would you know, basically we're basically identifying this molecule that we know associates with the AMPA receptors somehow. We think it must be doing something at synapses to alter plasticity. And so you can use a variety of behavioral experiments to determine what are the deficits that these mice have. For all practical purposes in the cage, they looked normal. They were able to breathe. They were able to survive. But again, in the cage, they didn't have, mice don't have to think that hard because they have water, they have food in order to survive. But you can test their ability to learn with a variety and remember with a variety of techniques. And this is where we collaborated with Jackie Crowley and her colleagues at the Mind Institute. So one of the classic experiments you can do is called the Morris water maze, after Dr. Morris, but and it's a little confusing because it's not really a maze. In this technique, you have a tank of water that is opaque, so you can't see the bottom. And within this tank of opaque water, there's a hidden platform. It's right under the water level. And this is it's a quite large tank, and you have it in this room where you have these visual cues surrounding the tank. And you put your naive mouse into the tank, and mice don't want to be wet. They want to escape. And so they will swim, searching around the tank in order to try to find a way out. Eventually, they'll find the hidden platform and they'll be able to be above the water. So this is good. Then you do this multiple times, multiple trials in one day and multiple days. And then after about 5 days, the mice actually get pretty good at this task. So you put them in the same tank. They will orient themselves to the cues that they now remember. Okay, the platform is opposite from the green circle. And you can measure this like as shown here. So this is called the learning phase, the latency to find the platform. And so wild type mice in the squares, the first day before, in the early training days, it takes them about 60 seconds to swim around this tank and find the hidden platform. But then over the course of 5 days, they get really good at this. So they less than 20 seconds. And this is robust learning. In contrast, the knockout animals that we generated could never really learn this task. It was as if they were doing this task for the first time every single day. So you see there's a slight reduction, but they were never able to really learn. They got a little bit better, but not at all like the wild type. And you could probe this learning by doing what's called a now trying to move I need to move this. Okay. We could probe the learning by doing this test here by removing the platform and then quantifying how much time the mice spend in the target quadrant. And so in this case, there's a target quadrant, there's the left, right, and opposite quadrant, right? So target, left, right, opposite quadrant. And wild type mice, again, spend the vast majority of their time in the target quadrant because this is where they found the hidden platform last time. Whereas the knockout mice spend equal amount of time within each of these quadrants. So again, they're not really learning. Even this decreased latency is not reflective of memory, contextual memory in this case. So this was quite striking. In order to validate, rigorously validate this finding, we performed an additional test of cognition, again with Jackie Crowley's group. So, this is not, it's a little fuzzy here. So again, this is called the novel object recognition task. So mice, if you give them 2 objects, they will spend equal amounts of time sniffing and exploring the objects. So this is the left and the right, the left and the right. And then if you give them a novel object, something they haven't seen before, again, the wild type mice will then spend the majority of their time with this novel object, because mice like novelty, and they'll remember they hadn't seen this one before. But again, our knockout animals treated the novel object as if it were like the previous object. So again, demonstrating these animals weren't able to learn and remember. So this is behavior. This is at the level of the whole animal. What's happening at the circuit level, and then ultimately what's happening at the synaptic level. So in order to look at the circuit level, we can use electrophysiology, and this is where we collaborated with my colleague Johannes Hell and a postdoc in his lab, Lucas Matt. So here's a schematic of the hippocampus. A major advantage for this structure is that there's very clear and stereotypical connections that you can follow. So the CA1 pyramidal neurons will form synapses with Schaefer collaterals from the CA3 region. So if you were to put a stimulating electrode here and then record from the postsynaptic structure, you can assay how strong this connection is here. And if you look in wild type animals, if you give a particular stimulus, this will lead to this long-lasting potentiation. This is called long-term potentiation that I mentioned. So they get this stimulus right here, and now their synapses have been increased by 25%. So they're now at 125%. And in the knockout animals here, you see, again, the starting at 100%, the stimulus is received here, but you see there's no potentiation. If anything, you might even see a slight reduction, depression. So this was also quite striking. So here we're getting a deficit in behavior that is correlated with this deficit in neural circuit plasticity. What about the individual synapses? So again, using our dissociated hippocampal culture, can we look and see if there's deficits in these synapses? And to do this, actually, let me see. Oh, yeah, let me just skip ahead because it's already starting, already 4. So to do this, what a graduate student in my lab, Lindsay Kirk, did is she utilized this dissociated culture and then looked to measure the effects on the AMPA receptor subunits. This is an antibody against one of the subunits, GluA1, in wild type and mutant animals. And then we can look at vGlut1, which is a presynaptic marker, and then look at the merge. So if you see, so here's GluA1, which is in red, and vGlut1, which is in green. So in the wild type, in the merge, where you see yellow is where there's a synaptic connection because you're getting pre and postsynaptic overlap. But you might also notice that there's a large amount of red in these neurons, and this is the reserve pools of AMPA receptors that are found outside of synapses. If you recall from the original diagram, there's AMPA receptors at the synapse, but there's a lot of AMPA receptors outside the synapse. And in our mutant animal, it turns out that the synaptic AMPA receptors were largely unchanged. So you can still see the presence of these yellow puncta, but it's this non-synaptic, dendritically or non-synaptically localized AMPA receptors that are missing in the knockout. And Lindsay was able to demonstrate this reduction in this extrasynaptic and actually an increase in synaptic GluA1. So this would be evidence that, again, the idea that SynDIG4, which is this red squiggle, is somehow keeping these receptors outside of the synapse, but only during LTP is it now allowing the receptor to be trafficked to the synapse in order to strengthen them. And this is needed. This is needed during the plasticity, and this is needed for cognition, is our model. And as I mentioned at the beginning, this is an example of a confocal microscopy. So the overlap, even though these structures are found in different cells, so the presynaptic versus the postsynaptic, these are two independent cells, but because of the resolution limitations of confocal microscopy, this appears as an overlap signal. So here's an example where you have bassoon and Homer, two types of pre and postsynaptic proteins. In confocal microscopy, this is why they appear yellow, because of the resolution. But you can use a super resolution microscopy in order to distinguish these now. So here's the bassoon, which would be here, and the Homer, which would be here. Now you see this very nice separation that you couldn't see with confocal microscopy. And this is an area that we've moved into more recently, the super resolution microscopy, and applied that to our system. And so, for example, here's a wild type animal or a knockout animal. Here's the glutamate receptor subunit, and here is one individual synaptic contact. And we can see not only the synaptic GluA1, which overlaps with the presynaptic marker, but this large pool of non-synaptic GluA1 that doesn't interact with the presynaptic, that doesn't overlap with the presynaptic marker, juxtapose, I should say. And in the knockout animal, as mentioned, the synaptic GluA1 appears normal or even increased, whereas the reserve pool, the backup GluA1 is missing. So this is, again, consistent with the idea that the SynDIG protein is somehow restricting the AMPA receptor from being trafficked to the synapse, and potentially only allows this to happen during times of long-term potentiation and other synaptic plasticities that would be important for memory. So for instance, when the mice are learning the task in the Morris water maze, perhaps the SynDIG is now less active or less able to retain the AMPA receptor outside of the synapse, and it can then be trafficked to the synapse, strengthen the synapse so that the animals can learn and remember where the hidden platform is. That would be our working model. So I know we end at 4:30, but I wanted to have more time for discussion. I'll just mention really quickly. So one thing that's been interesting is that the SynDIG proteins, like many proteins in biology, right? Proteins belong to families because they've evolved from a common ancestor. And so the SynDIGs belong to a superfamily that includes another group of proteins called the IFITMs. And this stands for interferon-induced transmembrane protein. And what's interesting about this group of proteins is that there's been, especially during the huge effort in viral research during the pandemic, these molecules are increased during viral infections. So these IFITMs are antiviral proteins. So these IFITMs are normally found in intracellular compartments. And when a virus, such as the coronavirus, attaches to its target cell on the plasma membrane, it gets endocytosed and then transferred to endosomes. And when it is transferred to endosomes, it will fuse with the membrane of the endosome, release the viral genome, and go on to make additional viral particles. However, when the organism senses an infection, it will increase the levels of IFITMs found in cells. These proteins are localized, as I mentioned, to these endosomes. And what they do is they inhibit the fusion of these viral particles from fusing with the target endogenous membrane, and therefore they prevent infection, or at least inhibit infection as best they can. And so this was really intriguing. And so other family members also appear to regulate membrane fusion in some way, right? So the question is, how are they doing this? These proteins have really unusual structures. So they're predicted to have this very long transmembrane domain that actually likely is at an angle because the helix is longer than the lipid bilayer. But they also have this amphipathic helix that's proposed to lie on the inner leaflet of the biological membrane. And perhaps this organization, which is shown here, is somehow inhibiting the ability of this fusion pore to form between a viral particle plasma membrane and a target cellular membrane. There's also other potentially regulations that we've been focusing on, but I want to emphasize what's interesting, right, is the fact that these molecules as a whole seem to promote or inhibit membrane fusion. And so how are the SynDIGs regulating AMPA receptor and synaptic strength, and are they doing this through some sort of membrane fusion regulation step? So what we've been trying to understand is how are the AMPA receptors, where are they localized? How are they affecting trafficking of the AMPA receptor during plasticity? And are they acting like these IFITMs to inhibit or regulate membrane fusion in some way? And let me see. 410. I don't want to go. I don't want to talk like just too much. Should I take questions here or have more of a discussion?
Yes, let's have a discussion.
So this is where we're focusing our efforts in terms of trying to understand how the SynDIGs are regulating membrane fusion, and then how that translates into its ability to change the localization of the AMPA receptors during plasticity, right? So perhaps, as I mentioned, there's this intracellular pool of receptors that's necessary during plasticity mechanisms, and that's what is regulated by the SynDIGs. And then under certain conditions, it'll either promote or inhibit membrane fusion to allow the delivery of the AMPA receptors. I think I have a slide. I have more data, but let me just show, right? So perhaps here the AMPA receptor, the SynDIGs are acting through mechanisms to either promote membrane fusion during plasticity. Sorry, I'm pointing at the right one, right? So the SynDIGs promote endocytosis to allow the receptor to be delivered to the plasma membrane, but perhaps they can also then inhibit endocytosis, right? So if you inhibit endocytosis, that would also lead to more AMPA receptors being stable at the synapse. So perhaps it actually is doing both, promoting and inhibit. And what's interesting, again, going back to this concept of the IFITMs, is that it turns out that depending on where the protein is localized, it will either promote or inhibit membrane fusion. So for instance, as I mentioned, normally these IFITMs are found within endosomes and they block fusion of viral particles there. But if you engineer them so that they're on the plasma membrane, you actually promote endocytosis, and infection is enhanced. So it could be that where the protein is localized will either allow for blocking of endocytosis or promoting of membrane fusion. So it could have this dual role, like these IFITMs are found. So I'll just end by thanking the members of my laboratory. This was at one of our recent outdoor, for the first time, in-person lab lunches that we are now able to enjoy. One thing, it's always been a pleasure mentoring students. There's always been at least 2 to 3 grad students in the lab, undergraduates in the lab. And then last year we hosted a post-bac student who's now a first year BMCDB student here on campus. So overall, and then longstanding support from Dave Specka, who is a research scientist in the lab, especially as all of us become more busy with various teaching and service activities. Knowing you have a key person that can keep the lab running is enormously beneficial. So thanks. I'm happy to answer questions or have more discussion. I really appreciate any suggestions or insight you might have. Oops. Oops. No. There we go.
Thank you, Elva. That was really interesting.
Oh, Maria, there you are. You made it.
Yeah, I'm here. I got here 2 minutes late, but I made it.
Oh, okay. That's great. Okay, cool. Oh,
Yeah,
I'm
no,
glad you could hear it then.
Yeah, so I'm not a neuroscientist, but I work a lot with viruses. So I was interested in that connection at the end.
Right,
And
yeah.
so these proteins, IFITMs, can either promote or inhibit the viral infection. So, I mean, is there evidence that, I don't know, that the virus leaves some sort of marker throughout evolution that will allow it to bypass any in the context of what you're studying? I mean, I still don't understand enough how these proteins work.
Yeah, so it's interesting. So it could be directly related to viral. There are infections in the brain. And so the SynDIGs are brain specific. They seem to be expressed only in the nervous system, but there are infections in the brain, and perhaps they also play a role in brain infections. That's one possibility. Or perhaps they evolved from a mechanism in the brain that was developed, that evolved to try to combat brain infections, and then the SynDIGs deviated and gained this new role in regulating AMPA receptor glutamate transmitter trafficking. So there might not be any direct relationship to viruses for the SynDIGs anymore, but we don't know.
Yeah, that's very interesting. And then on the just thinking about the viruses themselves, so the body has this mechanism to prevent the infection, to prevent the virus from being absorbed into the membrane. Mm-hmm. So is there evidence that the viruses evolve a mechanism to counter that? And if so, are those proteins interacting with the protein, these
yeah,
the FITM proteins?
Yeah, that's an interesting question. I'd have to, because again, the past few years, there's been this explosion in viral research, of course, due to the pandemic. So there've been a lot of recent, like hundreds, if not thousands of papers on anything related to viral infection. So there might actually be information about that we haven't followed up on to see, what are the countermeasures that the viral has developed, and then are those mechanisms also shared by the SynDIGs, and maybe there's some interesting informative things that we can learn that would help us understand their mechanism in plasticity.
So Elva, so do you know if these SynDIG proteins are altered in any neurodevelopmental diseases?
So the PR, so one of the related superfamily members is a disease-causing mutation in a human movement disorder, so a paroxysmal dyskinesia. The SynDIG4 that we've been studying, there have been genome-wide association studies with schizophrenia, but not, as far as we know, not disease-causing that we know of.
So, and in this neurodegenerative disease, this motor disease that you are talking about, you know if there is an alteration in the number of spines or synapses? There is.
Yeah.
Because then
there
you may infer that perhaps, I don't know, in autism, that they have also an alteration in the number of spines, that maybe the SynDIGs are a protein that we should look for, right?
Right. No, that would be really interesting. Yeah, there's a lot of avenues that, again, with more time or colleagues or collaborators, right? That would be really interesting.
Yeah. So we have a question from Lillian.
I actually have two questions. One is that that model that you just last presented about the forming like a kind of vesicle with the nuclear pore, with the pore in the membrane, seems very similar also to the formation of caveola. Have you look at if the SynDIG4 is also related to caveola formation, and if that will facilitate then the recruitment of the AMPA receptor?
So that's a great question. So these molecules have, let me go here. In addition to this structure, they also have cysteines, two cysteines that are found here that are reversibly palmitoylated. And it's known that the palmitylation of transmembrane proteins is one mechanism whereby you can get incorporation into membrane subdomains, such as caveolin, right? So we haven't looked specifically with the caveolin itself. We do know that they are modified. They have this lipid modification, but we haven't looked specifically at overlap with caveolin domains.
Yeah, because I remember reading papers about, especially caveolin-1, that deficiency in caveolin-1 could cause, deficit in memory. So I don't remember it. Yeah, so probably that would be kind of a good avenue to explore.
Okay, I'm writing this down. That's a great suggestion. Thank you.
Yeah, and then my second question was related to the viral infection aspect, since it's known that long COVID is associated with what they call the memory fog, COVID fog or COVID brain. Well, there are very different names for that. That probably, precisely that it could be an interaction of COVID infection with this process that is probably the culprit of the reason for why people are having the memory fog that they associate with long-term COVID.
Yeah, that's really interesting. I think as there's more research now being done on the long COVID, because again, the mechanisms for that are not clear, right? But I hadn't even thought about that. So maybe there's this, actually, it could be sort of related to what Maria was saying, that if maybe over time, the virus is able to overcome our defenses, right, in order to lead to this long COVID. And maybe that's partly related in some way to this viral infection within or alterations, perhaps, in this type of model. I hadn't thought about that, but that's really, yeah, that would be really interesting. Thanks, I'm writing that down too. Really good suggestions. Yeah, we need to, my plan, like everybody, we have to start writing grants, but yeah, that I need to start writing more grants this year.
Yeah, and I think that at the Primate Center, they did a couple of experiments with some rhesus macaques that were infected with COVID. So probably there is still a reservoir of those brains that could be looked for immunohistochemistry.
Oh, right. Yeah, and just see if there's alterations or upregulation. Yeah. No, it's a great idea.
You can look also in the human brains. I think that I have a couple of brains with COVID.
Oh, yeah. Okay.
Yeah, I normally don't take them, but it's okay with that.
Yeah, that would be really, I hadn't even thought about that. That's a great, because again, if we observe any sort of change, that would be an interesting avenue to pursue to try to link between the COVID brain, the brain fog, right? And overcoming this infection or overcoming the defense mechanisms that the cells normally have.
Yeah, and even in the case of HIV, that it's true that it's a different type of virus than the coronavirus, but also people that have been, even with the antiviral, the ART therapy, long term, this called also causes neuroinflammation that is associated also with cognitive dysfunction that probably would be associated the same way with this type of mechanism.
I see. Yeah, fascinating. Yeah, I think, we've definitely read a lot more of the viral literature because there has been a lot more, and then we saw this connection. But yeah, a lot more direct connections might be a really interesting avenue to go. Yeah.
Okay, next talk.
Okay.
Next question.
Yeah. No worries.
You have more questions?
No, I don't have myself, but yeah.
I might follow up because those are really good suggestions. I hadn't even thought. But yeah, if we can observe any alterations, that would be really interesting. Yeah. Cool.
Well, I guess
All
that
right.
looks like we are done for today.
Yeah.
Thank you so much, Elva. This was a terrific talk, and I really enjoy also your personal experience and your travel.
Yeah.
So this was really good, and we need to do more of that.
Right. Yeah. We build our community.
Really, really interesting. And I think that, yes, that I made your mom once.
Oh, good. I'm glad. Yeah. It is sort of interesting. She's is still, such a huge part of my life that many people have met her, many of my colleagues. And I don't think I've met anybody else's mom, but I'm like, it's like, so I think it's very, especially in our culture, right? It's very important. So
Yeah.